Hello, everyone. I'm Edwige Ponseel, Dystonia Europe President. Today is Monday, June 9, 2025. I'm glad to introduce you to Dr. Ahmet Kaymak from Italy. Hello.
Hi.
Dr. Kaymak is our Dystonia Europe David Marsden Award 2025 winner. Let me congratulate you for this award on behalf of Dystonia Europe and the Dystonia community. Would you like to introduce yourself?
Yes. Hi. Hello to community. I am Ahmet Kaymak. I am originally Turkish. I have been conducting research in Italy at Biorobotic Institute in the Computational Neuroengineering Laboratory of Scuola Superiore Sant'Anna in Pisa. Yeah, I have been researching about the neurophysiology of movement disorders, mainly in dystonia. I have been working for understanding the dynamics of dystonia for more than 5 years. So that's basically my background.
Good, thanks. So you have won the David Marsden Award for your paper, Spiking Patterns in the Globus Pallidus Highlight Convergent Neural Dynamics Across Diverse Genetic Dystonia Syndromes.
Perfect.
Would you mind explaining your subject to the patients community we represent so that they can easily understand your study?
Yeah, I'll try my best. So this work basically focus on understanding dynamics of really complex disorder. When you look for the other disorder, the origin of the disease generally, can stem from one thing. For example, in Huntington, there is one gene. If there's a pathogenic mutation, you will have the disease. But dystonia, there are multiple brain area come together to create this disorder, and the genetic background is extremely rich. And when you check each gene function in our brain, they have widely different function, but they somehow creating the same disorder. And we'll try to understand how this pathogenic mutation affect the neural behavior in the part of the brain that control our motion, and if we can learn from these pathogenic changes in the neural behavior to see that why some patients with specific genetic condition respond well to the brain stimulation treatment while the others are not. So
Okay,
that was basically the summary.
okay. Do you want to share something and
Yes,
explain more
yeah,
in details?
I prepare some slides.
Okay,
So just 1 second, please.
you're welcome.
Um, Share the screen.
Yes, it's this one.
Yes, start now. Perfect. Just please let me know when you start to see my screen.
Yeah.
Okay, perfect. Okay,
Good.
I will. Yes. So, hi again. Today I will present my research about what I said previously, and I try my best to make it as clear as possible. So, I will start to introduce the movement disorders. These movement disorders are the group of neurological condition that leads to dysfunction of our motions. And it can be either hypokinetic or hyperkinetic. In hypokinetic cases, we are seeing like the motions and the movements tend to slow down. But in the hyperkinetic cases, like in the dystonia, the activation of muscle more than normal. Um, and there is particular part of brain is generally considered for the origin of this disease. It is called basal ganglia. It is this structure is part of our motor circuitry. This motor circuitry you can think as a computer circuit that really specialized for a particular function. The motor circuitry basically plan and execute our motions. And it'll basically send signal to our peripheral system, the muscle, that we can do these encoded motions. I would like you just to remember this part, the globus pallidum, and this is a substructure within the basal ganglia. Just this one, globus pallidum, to remember. As we already know, the main therapies for dystonia, the pharmacological drug treatment. In some cases, also the patient gets the botulinum toxin, the Botox injection to the muscle that's showing the dystonic symptoms. But if the dystonia is a type of dystonia resistant to these drugs, drug treatment, the neurological teams and neurosurgeons can suggest the patient to go under the deep brain stimulation surgery. But what is this surgery? In this surgery, there will be electrode placed part of this motor circuitry. In this case, dystonia, mainly globus pallidum, and we will provide electrical stimuli to this part to basically suppress the pathological activity to restore our normal motor function. But when we talk about this structure, they're really small. They tend to their length tend to go from 2 millimeter to 10 millimeter, and it is highly hard to target them. For targeting them, the doctors generally use a specific technique, microelectrode recordings. In this technique, they basically place an electrode to part of that brain and would like to see a really distinct behavior to see that, okay, we are in this target area. So what this electrode showing? Around the tip of electrode, we have basically neurons. They function as themselves, and we get their activity like this as a raw recording. What we analyze in this research to this raw recording to get activity of single neurons. So we have the activity of neurons affected by the dystonia itself. This is extremely rare recordings, and we would like to analyze it to see if we can understand anything from pathophysiology of it. So this is the activity that we try to characterize for each gene in this work. Coming back to genetic dystonia is super complex because there are now more than 50 genes that shown conclusively pathogenic, leading to dystonia, but there are more than 400 genes broadly associated with the dystonia. And when you check the biological function of this gene in our brain, they're completely different in some cases. In some cases, they more focus on signaling in our cells. In some cases, they involve the energy production. The big question is that how they can generate the same disease if they have really diverse function. When we talk about dystonia, it can be dystonia, but the patient experiences dystonia in really different ways. Here are the four genes: TOR1A, THP1, GNL, KMT2B. I basically showing this because these are also four of the 9 genes that I also analyze. As you can see that the type of dystonia significantly change from gene to gene. For example, KMT2B patients generally tend to have like a complex dystonia compared to this other 3 group. Even like at the initial symptoms, the body part affected by the dystonia at the onset of the symptoms can significantly different. For example, for GNL cases, there will be cervical dystonia around the neck, but for DYT1 or 1A cases, it'll be in the limbs. For THP1, it is more heterogeneous. Even the progression of these symptoms can differentiate from gene to gene. For example, for GNL, symptoms tend to localize around the neck, cervical dystonia, and it won't spread to the other body part. But KMT2B, TOR1A, they tend to generalize in time. This is why I'm showing, even if it's the same disease, the type of gene can determine what kind of symptoms in which body part affected by the dystonia itself. One of the differences between these genes also responds to the DBS treatment itself, because genes like TOR1A, GNL, KMT2B patients, they really gave good response to DBS treatment itself. But when it's come to THP1, the intended therapeutical benefit is much more limited, and the reasons why is not super clear for us. In our research, what is also, we basically, let me start to give you some background why we try to decide to go on with this project. Because what we're seeing that even the dystonia genes have like a different biological function, they eventually leading to the same diseases, and there is convergence, common behavior at the different anatomical scales within the cell, from different cell type. And for example, the last year's winner of the David Marsden Award, the different activity level in the part of the brain, basal ganglia. So what I'm saying that even their function is different from intracellular to system-wise anatomical structure in our brain, they start to converge functionally. And there were actually two other reasons that we want to investigate this, because neural activity related to potentially common behavior among dystonia genes at motor circuitry level that control our motor activity remain largely unexplored. And in addition to this different clinical manifestation, the symptoms, the neural mechanism potentially connect to why some forms of genetic dystonia syndromes respond well to DBS treatment, while some others, there is the therapeutical benefit is limited. And here are our research objectives. First, we try to characterize the gene-specific neural activity for each dystonia genes in our patient group, and we try to group them with the advanced analysis. Because if we can group them, we can check if the same group also align to the DBS treatment outcome itself. This is important. And we try to investigate the connection between the neural activity and the DBS outcome across our genetic profiles. And we would like to compare the established finding in the clinical literature. I told that for TOR1A GNL patient in the literature, they respond well to DBS treatment, but THP1, it is not. So if we can see that, we can validate also our finding with the clinical literature and also conduct open science practice. We really aware that the recordings that we are analyzing, they are really rare. And to do this reason, to also serve the community, we make sure that all the data is publicly accessible. All of the codes, all of the machine learning models can be accessed by the other group to make this research more accessible for the other group if they want to build on top of our research. So basically, I will give you briefly our experimental design. In our cohort of patients, we had 31 dystonia patients having pathogenic mutation in these 9 genes. All our patients undergo GPI DBS surgery, and we collected this electrophysiological recording when they're under the surgery with the same surgical procedure to make sure that the data is comparable between different patients. Then the hefty process came for more than 8 months. I analyzed more than 300 recordings, and from these recordings, I successfully get the activity around 1,700 neurons. And for each neurons, I measure set of, let's say, future metrics to characterize how these neurons function. I will show you some results to make them a bit more clear. If the neurons function regularly with the pauses irregularly. So I try to characterize that. This is a reconstruction example that I did in another paper. As I told you, let's remember the globus pallidum. Here you can see the 3D reconstruction, and you can see the trajectories of this recording in the real life. And we analyze this recording that reside within the globus pallidum. The part targeted by the DBS to be stimulated. Okay, I will show you something super brief to make you better understand what kind of thing that we try to analyze as a neural behavior. For example, this is a neuron. You can see the activity. Each line here represents the neuron spike, neuron work for something, a unit activity. And you can see that within the time, it was relatively regular. I am saying relatively compared to an irregular neuron. So here at the beginning of our recording session, the neuron was working a lot here, but in the meantime, the activity decreases. So this is we deem as irregular. And in the other third case scenario, we are observing some neurons work really hard in short time period, followed by pauses. As you can see here, the activity decrease to zero, increase again, decrease to zero, increase again, decrease, increase, decrease. This is like a bursting behavior. So what we do is basically check that the fraction of neurons among these 9Gs showing particular behavior, if they are working regularly, we're calling tonic. If they are working irregularly or bursting behavior, even when you check it, visually, you can see that the genes start to group here. So, for example, these three genes, THP1, PENG2, and AOP genes tend to show like a more regular activity. In the meantime, SGC, GNL, KMT2B neurons tend to be more bursting behavior. This is like a visual confirmation. And then this is, I know, a complicated graph. All I can say here in this axis, you can see weird name neural metrics. And in the y-axis, you can see gene pair. Each dot here represents if the neural metric is different between this gene pair. So even when you visually see that some gene pairs almost show identical activity compared to the others. So we can visually, again, group the genes by checking the differences between each metric that I try to characterize the neural activity. But we need to be more scientifically proved. So we developed this technique to make sure that it is statistically supported. In this figure, I would like you to focus only this part, the tonicity means the regular activity. As you can see here, these three genes, PENG2, THP1, significantly show high degree of regular behavior compared to these 5 genes, the 6 genes. So, and we can significantly separate it. So we have 2 groups, but you can tell Mohammed what these mean represent. So let's remember that these 3 genes show really regular activity, these 6 genes not. This is a good review. So I would like to tell you how DBS work. DBS basically work by desynchronizing the pathological activity. So compared to the non-pathogenic brain, we don't see the pathological activity increase. So what I'm saying that, for example, for Parkinson disease, when we investigate another part of the brain in a particular frequency band, there will be higher activity. So when patients have like a Parkinsonian symptoms, this activity increases, and DBS try to decrease this activity. But for dystonia, the frequency range is theta alpha. When patients show dystonic symptoms, this activity increases. So DBS try to remove this activity, but how? How? As I told, in dystonic state, we have synchronization, increase activity in this frequency range, and we give stimulation. And then from dystonic state to symptom-free state, this pathological increased activity will be gone. But what our hypothesis is that this pathogenic activity can appear with a bursting behavior. So what we are saying that if the genetic dystonia syndrome, the gene tend to produce neurons with regular activity, this activity may not appear, which DBS can decrease and restore the symptom-free state. So that's why our grouping says that if the neural behavior bursting, DBS can work better compared to the regular activity. So this is the main takeaway. But we have limitations. We have comparably small patient cohort. Because why? Due to the rarity of genetic dystonia syndromes, this is good for the patients because these genetic syndromes really rare. In our study, we had limited number of patients, and as I told you, there are more than 50 genes that conclusively associated with dystonia, but we only analyzed 9 of them. And our findings are not causal, but correlational and exploratory. So there are a lot of things that limit our ability to draw 100%, certainty for our results. Why? Some genes, active in the other part of the brain compared to the globus pallidum. Our observation regarding the neural behavior, correlational, not causal. And in some cases, there is the pathogenic mutation in the person, but the patient won't have the dystonia itself. This is also something interesting. So that's why we are telling that we need more research to validate our results. So how? We need to replicate this study with more patients and including with more genetic profiles to have like a more stronger results. And in our research, we collect the recording during when patients under anesthesia, but we need to replicate this study also in different conditions. First, they should be in awake resting condition while they're conducting different type of movement to characterize if the type of the movement also affect the activity, the neural behavior itself. If the patient take the pharmacological treatment or not, the drug on and off condition, and also if the stimulation, if we are delivering the electricity to that part of the brain or not, we need to characterize the neural behavior in all of these different conditions to better aim regarding the DBS treatment itself. So I just briefly would like to thank you, my professor advisor, Alberto Mazzoni, and we have the smartest and kindest neurologist in Carlo Besta Neurological Institute, Luigi Michele Romito. I would like to thank them to give me a chance to collaborate with them. Thanks a lot. This was my presentation for you.
Tremendous.
Sorry.
Yeah.
Okay.
Can maybe stop the screen
Yes,
share.
I
Yeah.
just, yeah.
How long did this study take?
4 years.
4 years. Oh my
Yes,
God,
because
tremendous.
the thing is that some of the recordings that we already had and some of the patients during my PhD, we took the recording, but the main challenge was, to get the individual activity of this neuron took a lot of time. And it really takes a lot of time, and also make sure that we are not seeing the other activity of the other neurons. It took a lot of time to validate. So it's also, we make sure that the things that we are saying is scientifically, supported to interpret the results, because there are multiple dimensions. I am not geneticist. I was sending a lot of email to clinical geneticists. I was getting information about the electrophysiological experts, the clinicians. I was doing like the machine learning, the statistical analysis. Basically, there were a lot of puzzle pieces to put. It took a lot of time, yes.
And yeah, of course, the question from your point of view, do you think it will be possible in the future to have this second step of the study with more patients, with different
Yes.
conditions, etc.?
After the publishing of this paper, we get some, of course, the collaboration request. But in the meantime, I already have also a couple of papers already in production. So we will publish like a support the finding. We are also planning to conduct more international collaborations, also the organoid studies. Also, one of the things is that these recordings that we are showing only the activity of single neurons.
Mm-hmm.
That is not, seen by the DBS electrode, because DBS electrode saw like a couple hundreds of activity of neurons. So we need to make sure that the differences that we are seeing also reflected to the population of neurons. And we also, for that, we are planning to conduct some research to really, push forward in this genetic dystonia treatment. Hopefully, we will be successful.
Hopefully, great, great, great. So thanks, Dr. Kaymak, for this lecture. We hope this award will encourage you for continued research in the field of dystonia, as we are always happy to see young researchers who have the interest and passion to move dystonia research forward. We thank all of you who have listened to the presentation, and many, many thanks, Dr. Kaymak.
I also would like to thank, first of all, the Dystonia Europe. It is also for us important that the work is, like a find worthy by the actual patients that, causes also the board. This is important because we are now sure that our works resonate. It won't only stay in a paper, but can also find interesting by the patient themselves.
Mm-hmm.
I would like to thank you also for this opportunity to discuss and connect with the community. Thanks a lot.
Thanks to you.